Good morning, everyone. This is Elaine Hu from Bristol Myers Squibb. Today, I will be the moderator for the webinar. It's my great pleasure to introduce Dr. Amelia Curtin to be the invited speaker of our today's IQ Nanotechnology and Nanomaterial Working Group webinar. His presentation today is about oral delivery of nucleic acids. Dr. Amelia Curtin is an instructor in medicine at the Brigham and Women's Hospital, Harvard Medical School. He received his undergraduate training in pharmacy in India before moving to the US for graduate school. Later, he earned his PhD degree under the guidance of Professor Jayant Panyam at the University of Minnesota and carried out his postdoc training under the supervision of Professor Bob Langer and Professor Gil Traverso at MIT. He has co-authored 25 research articles and has been a co-inventor on 6 patent applications. Dr. Curtin is passionate about using pharmaceutical engineering principles to solve the most pressing medical problems and in training young scientists in this discipline. Without further ado, I will turn the floor to Dr. Curtin for his presentation.
Thank you very much, Elaine, and I really want to thank the organizing committee for giving me the opportunity to be here and to share my research with you. Before I get started, I kind of wanted to give you guys a couple of disclosures. So the first one is that my training is as a drug delivery scientist, and I have just recently started working with nucleic acid based therapies, and so I'm still learning this field, and there's a lot more to do over here. So hopefully what I'm able to present to you today is something of interest to you and something that can be useful to you going forward. The second sort of disclosure here is that our interests over here lie on the oral delivery of mRNA. As you can imagine, this is it's a really complex field, and there's very little work done on this. So what I'm going to present to you today is really one of the first steps taken towards achieving this goal. The data that I present to you today is really proof of concept data. This is not a product that is ready to go out in a person today, but hopefully the investigations that we have started over here lead to sort of more engineering and scientific discoveries that can ultimately lead to something that can go into a person. So that all to say that both myself as well as this particular field are in the relative infancies, and so there's a lot more to learn over here, and I hope what I present to you today is of interest to you. All right, so before I get started on this specific topic of oral delivery of nucleic acids, I just wanted to give you a little bit of background on myself and some of the training that I've had in the past, and this will sort of explain to you why we were working in this area. So as was said in the introduction, my training lies in drug delivery, and specifically 2 aspects of drug delivery. I did my PhD work with Professor Jayant Panyam at the University of Minnesota, where I worked predominantly on formulation development, really focusing on nanoparticle-based therapies for targeting small molecule drugs, chemotherapeutics to tumors. This was mostly based on polymer-based nanoparticles. For my postdoctoral work, I switched over fields a little bit, and I started working on device development, and we specifically focused on making devices that can be orally ingested. So these are devices that can go into the gastrointestinal tract, and this work was done with Professors Bob Langer and Gio Traverso. So with these 2 sort of skill sets, we've used it towards different applications. The first one has been for sustained small molecule delivery. Of note here is that we we developed a pill that is orally administered, and that can give you sort of drug concentrations out to a week or out to a month, and we showed sort of proof of concept of this in large animal models. We've started some work on macromolecule delivery, which is obviously what I'm going to talk about today. And then sort of the third application of formulation development and device development has been towards cancer therapeutics. I won't talk much about this today, but if there's interest, I'm happy to discuss this later. But this is where I came from, and this is where we started sort of working on mRNA-based therapies. So there's rightly a lot of interest in designing mRNA-based therapies today, and there can be multiple reasons for that. I've just listed a few of them over here. So as you know, a majority of the drug products on the market today are still small molecule-based drugs, and they perhaps might remain a majority of the drug products in the market. But nonetheless, small molecules sometimes have limitations in that you may not be able to target all of your proteins of interest with small molecules, perhaps because of the folding of the protein, perhaps because of the physicochemical properties of the nanoparticle, of the small molecules that you develop to target those proteins. But in theory, nucleic acid-based therapies, such as mRNA-based therapies, can be used to theoretically target any protein of interest. So it gives you this sort of platform-based technology that can be used to modulate a lot of targets. mRNA-based therapies, by definition, are produced inside of a cell. So one has the potential to target intracellular proteins with this. In contrast, if you think about some other macromolecule-based therapies, such as proteins, at least what's in the market, a majority of these proteins target extracellular receptors. And so their ability to target intracellular targets is relatively low. So, but in contrast to that, because the mRNA is translated intracellularly, there's a lot of potential over there. A third piece over here is probably a little more specific to vaccine development, and certainly something that we are all thankful to today, which is the speed of development. And we've seen this with the SARS-CoV-2 vaccine as to how quickly these two vaccines were developed by a couple of companies who developed this. So certainly this has a lot of potential when it comes to sort of vaccine-based therapies. And then finally, as opposed to DNA-based therapies, which may have the potential to get integrated into the genome of the host, mRNA-based therapies have a relatively lower chance of that happening, and that's why they leave behind lower footprint. So all of these, and certainly there might be others, which I'm not talking about over here, has generated a lot of interest in producing these mRNA-based therapies. Now, as you start sort of thinking about these mRNA-based therapies, there are a lot of considerations that that one must think about. Certainly, the chemistry of the mRNA has a major impact on how these therapies perform. The choice of nucleic acid bases, this has been really at the center of why these mRNA-based therapies are useful today, and has garnered a lot of interest and attention of late. In addition to that, the purity of the mRNA that one is using in the therapeutic can also have a great impact. For example, there has been some recent work which talks about how removing double-stranded mRNA impurities from the mRNA can help you make an mRNA vaccine more tolerogenic as compared to more immunogenic in nature. Depending on the kind of immunogenicity and the length of expression that you need, things like circularization of the mRNA could also be thought about when you are designing these therapies. A second piece that that we think about, especially because we work very closely with the Gates Foundation, is the storage and stability of the mRNA. So as you're starting to think about using these kind of therapies in, say, low socio-demographic index countries where you may not have cold storages, how do you actually have these therapies get to the patient and still sort of sit around for a long time? So these are, of course, very important considerations, and one must really think about them deeply. But because of my background in drug delivery, my interests over here have predominantly focused on how do you actually deliver the mRNA to its site of action. And this figure over here on the right really does an excellent job, in my opinion, of showing all of the barriers that one needs to come across to get the mRNA to the cytoplasm where it can be translated. So once the mRNA is injected, typically it's injected today someplace within the body, either subcutaneously, intramuscularly, intravenously, or so on. It gets exposed to significant sort of concentration of nucleases, which can rapidly degrade this mRNA-based therapy and really make it useless. So finding ways to actually protect the mRNA while it's in physiological conditions is really very important. Now, depending on where the mRNA is administered and where it's going to act, it will need to get across at least 1 or 2 cell membranes for it to be actually useful. And because these mRNAs are macromolecules negatively charged, obviously their cell uptake is relatively low, and so finding ways to improve their cell uptake can be really very important. Other considerations relating to the immunogenicity, the reactogenicity of the mRNA itself, as well as the excipients used to formulate the mRNA are also some things that one needs to consider. So with all of this sort of multi-pronged problem, I should say that we've made significant progress in this field, especially focusing on the delivery of the mRNA. And over here is a snapshot of the materials that have been used for the delivery of mRNA. So on the left over here, I'm showing you a series of lipids that have been developed, and these are sort of obviously one of the most advanced delivery approach today for mRNA-based therapies. These are typically positively charged. They have nitrogens in their backbone over here, which allow you to conjugate or to complex, I should say, negatively charged mRNA to form the nanoparticles that allow you to then get the mRNAs intracellularly. But in addition to that, another sort of delivery modality that has been looked at has been polymers. Very similar to what is seen with lipids, these polymers also have a nitrogen present in their backbone, typically secondary or tertiary in nature. And because of the secondary or tertiary nature of the nitrogens in them, they have a cationic charge, and that cationic charge allows you to complex anionic nucleic acids, allowing for their intracellular delivery then. So there's really a lot of work done towards the delivery of mRNA as well. And with all of that said, with all of these sort of work on the chemistry of the mRNA and work on the delivery of the mRNA, we've had significant preclinical and clinical success with currently existing technologies. Of course, you know that these mRNA-based therapies have been widely used for vaccines. There has been a lot of evidence to show that you can have robust B-cell immunity and also some T-cell immunity elicited with these mRNA-based therapies. So obviously these can be and are being used for treating infectious diseases, but you can think about other kind of diseases such as autoimmune diseases and cancers that these can also be applied towards. There has been a significant amount of work done now towards using mRNAs for protein replacement. And for these kind of approaches, a lot of these mRNA-based therapies are administered systemically. And now people have shown that you can have this mRNA delivered really very well to the liver, but you can also have mRNA delivered to the lungs, spleen, bone marrow, endothelial cells. I'm sorry if I'm missing out a couple of organs, but these are sort of the predominant tissues that I have seen being targeted with mRNA-based therapies in literature. Very interestingly, people have shown now that you can also have sort of the biodistribution of this mRNA, or at least the expression of the mRNA, can be significantly changed by altering the lipid that goes into the nanoparticle, or the polymer that goes into the nanoparticles. So there's really a lot of very interesting work done in this area. And then finally, there is some work being done also towards local therapies with mRNA. The couple of them that have stood out to me is some work that's been done on local therapy within the eye with these mRNA-based therapies, and also, of course, a significant body of work that focuses on locally administering the mRNA in the tumor, and then activating downstream immune system to even get systemic effects with mRNA-based therapies. So all of that to say that we have learned a lot about mRNA-based therapies, the chemistry of them, how to formulate them, and we have had a lot of preclinical as well as clinical success with mRNAs. And of course, this is probably just the start to the field. There's a lot more to do going forward. But when we got started in this work, really the question that we wanted to look at can be summarized through this figure. We noted that a lot of mRNA-based therapies, if not all, are injected today. They can be injected at various sites within the body, intramuscularly, intradermally, subcutaneously, intravenously, but nonetheless, most of these are injectable formulations. So the question that we wanted to ask ourselves was, is it possible to actually convert this injection or an injectable product into a pill that a patient or a person can swallow to get therapeutic efficacy? And so really our goal was to try and convert this injectable mRNA therapeutic into an oral mRNA therapeutic. So what was our motivation behind doing this? So in my mind, at least, there were really three potential advantages of having an orally administered mRNA-based therapy. Then the first is that of access. So as I presented a couple of slides ago, most mRNA-based therapies, when they are administered systemically, they typically go to organs such as the lung, liver, spleen, and so on. To date, I haven't seen many mRNA-based therapies that are able to target the gastrointestinal tract. And so targeting diseases of the gastrointestinal tract, such as inflammatory bowel disease, ulcerative colitis, or allergies associated with foods can be somewhat difficult to do with these systemic mRNA-based therapies. But in contrast, if you were to deliver the mRNA-based therapy orally, so the gastrointestinal tract is the first pass, which is the part of the body that gets the maximum concentration of the mRNA. And so one can potentially see how orally administered mRNA-based therapies could be used for treating diseases of the gastrointestinal tract. A second advantage of this is that about convenience. So this might really differ from patient to patient, but several patients might prefer to take an oral pill over a systemic injection. So potentially having an mRNA-based therapy that is given orally could be preferred by several patients who do not like injections. In addition to that, most mRNA-based therapies typically are given by a healthcare provider. If you have a pill that can be potentially self-administered, so that could reduce the burden on our already burdened healthcare system. And then sort of very focused topic of vaccination. So today, mRNAs vaccines are obviously given systemically. And what has been learned, at least from the field of protein-based therapies, is that when you give a protein subunit or a subunit vaccine mucosally, you can potentially have a mucosal immune response, and you can also have a higher systemic immune response. So giving a vaccine in the gastrointestinal tract at a mucosal site could really help with that as well. So these were some of the opportunities that we were going after as we tried to develop these this orally administered mRNA therapy. But just as you observe with systemic mRNA therapies, oral mRNA therapies also have significant challenges. And these challenges are very innate to the gastrointestinal environment. So just as in systemic circulation, the gastrointestinal fluid also has a lot of nucleases present in it. So if an mRNA is ingested, it will be rapidly degraded by these nucleases. This sort of bullet point over here of pH is kind of interesting, and it's very specific to the gastrointestinal tract. So the pH of the gastrointestinal tract, as you go from the stomach to the rectum, really changes from pH 1 or so to up to pH 7. This pH changes during the day, and it changes from one patient to the other. So really, kind of the fluctuations that you see over the population are really very high in this kind of space. In contrast, I would think that the systemic pH across populations would be relatively narrow. So my point here is that designing formulations that are able to sort of work at different pHs becomes somewhat challenging with this. And then food. People take drugs at different times. There may or may not be food present, and this can obviously have an impact on how these formulations work. A second sort of challenge to delivering mRNA orally is that of access. So the gastrointestinal tract is lined by a mucus layer, and this mucus layer is this biophysical and this chemical barrier that can prevent macromolecules from diffusing towards the cells, from diffusing from the lumen into the cells. So really overcoming this mucus barrier to get to the site of action can be difficult. And then just as with systemic mRNA therapies, you still have to overcome biological membranes in order for that mRNA to be translated into the protein of interest. So, there's obviously several opportunities available with oral nucleic acid therapy, but there's a lot of challenges associated with this too. But despite these challenges, there has been no dearth of effort really when it comes to designing oral nucleic acid therapies. And over here, I'm just giving you a brief table that sort of summarizes some of the work that has been done in this area. Typically, what people have done is they have designed nanoparticle carriers. These nanoparticles by themselves have some stability within the gastrointestinal tract, or they put the nanoparticles into a secondary carrier, which gives them even more stability within the gastrointestinal tract. And they have used this for the delivery of antisense oligonucleotides, siRNA, and plasmid DNA. And this has had varying degrees of success, both in vitro studies as well as in some preclinical models. All of that to say that there has been a lot of effort in this area, but I should say that the examples that I came across, they were mostly focused on nucleic acids that were not mRNAs. Our focus over here was on mRNA-based therapies, especially for our in vivo studies. And this was a difficult challenge because mRNAs have even lower stability than some of what I've shown over here. All right, so this is sort of the summary of the project that we worked on. So as we were trying to develop these oral mRNA-based therapies, we were trying to overcome two barriers. Number one, how do we protect the mRNA within the gastrointestinal tract? And number two, once the mRNA enters the wall of the gastrointestinal tract, how do we get it across the cell membrane so that it gets expressed into the protein of interest? So in order to tackle this twofold problem, we came up with a twofold solution, which I'll be sharing with you today. I'll first address this problem of how do you get mRNA across the cell membrane, and then I'll go into sort of the first part of the problem, which is how do you actually protect the mRNA while it's in the gastrointestinal tract. Okay, so in order to deliver the mRNA intracellularly, I was interested in using polymers. This was something that I was trained in during my PhD, and as I'll share with you, there are a couple of advantages associated with these systems. And the specific type of polymer that I was interested in working on were these polybeta amino esters. So these poly-beta amino esters are formed by a Michael's addition reaction between an amine and a diacrylate. The diacrylate gets added on sort of at the place of the hydrogens of the amine and giving you the amine getting incorporated into the backbone of the polymer. Because you have this amine in the backbone of the polymer, these polymers are positively charged and they can complex negatively charged nucleic acids. The interesting thing about these polymers, at least to me, was that number one, just by changing the functional groups that are attached to the amine and a diacrylate over here, you can make a library of these poly-beta amino esters, so you can have a lot of chemical diversity built into your formulations. And number two, as you can see over here, that these polymers have an ester built into the backbone of the polymer. So these are biodegradable polymers, and in my hand, these polymers almost completely degrade in vitro in about 24 hours. So these can be eliminated relatively easily from the body. And a lot of work, and I should really stress, a lot of work was done on these polymers by Professor Dan Anderson and Professor Bob Langer in the early 2000s. So there was a lot of sort of known information about these polymers when we started working on this. But I should say that a lot of the polymers that were developed before were all sort of these linear copolymers that I'm showing you over here. Now, in parallel to this work on poly-beta amino esters, there was a lot of work done on this other class of polymers called polyethylene imine. So these polyethylene imines also are nitrogen-containing polymers. With this polymer, people had shown that branched versions of polyethylene imine were much more efficacious as compared to linear versions of polyethylene imine when it came to plasmid delivery. So taking these two fields together, we wanted to ask the question, would branched versions of poly-beta amino esters be better at delivering nucleic acids as compared to linear versions of poly-beta amino esters? And we thought that if we are able to get more potent transfecting agents, we'd obviously be able to deliver more of the protein with a smaller dose of the mRNA. So in order to sort of design these branched polymers, we really made a library of branched polymers. So we started off with the linear poly-beta amino esters, as I'm showing you over here. We made this such that there was an excess of acrylate groups in these polymers so that you end up with acrylate groups towards the two ends of the polymer. We then conjugated these linear poly-beta amino esters with what I refer to as branching agents, which are shown here in pink. So these are small molecules that typically have at least one primary amine, and then secondary amines are thiols. And the hydrogens of these amines and thiols can be reacted with, or the nitrogens react with sort of the terminal ends of the linear polymers to give us these branched polymers. So by changing the linear polymer that went into the reaction, or by changing the branching agent, we really synthesized a library of these branched poly-beta amino esters. So in order to sort of characterize this, we first wanted to understand whether this branching reaction was going forward or not. So to answer that question, we used two orthogonal assays. In the first assay, we were measuring whether the branching agent was getting consumed in the reaction or not using a biochemical assay. So over here, I'm showing you the percentage of branching reagent over time in the reaction. And you can see that over the course of 24 hours, the concentration of the branching agent reduces to almost zero, indicating that this branching reagent is getting used up in the reaction. To sort of confirm these results, what we did was we tried to look at the intensity of these acrylate peaks in the linear polymer using NMR. If the intensity of the acrylate peaks in the NMR went down, that was suggestive that you are getting the reaction going forward. So over here, I'm showing you NMRs for several of the polymers that we synthesized. The blue NMR signals over here are before branching, and the red ones are after branching. And you can see that these NMR signals from the acrylate peaks went down after branching. So this helped us sort of establish that indeed we were able to get these polymers to be formed. The next question was really going back to the hypothesis that we had, do branched polymers deliver nucleic acids better than linear polymers? And at the start of the experiment, we started to do these experiments with plasmid DNA. And for the first experiment, we used these raw cells, which are a mouse macrophage cell line. We conjugated, or excuse me, we complexed our plasmid DNA with our various polymers to form nanoparticles. These plasmid DNAs express GFP, green fluorescent protein, and so we could actually measure the expression of this green fluorescent protein in cells using flow cytometry. So I'm showing you over here the percentage of transfected cells in this cell line. Each of the circles over here is a different polymer. The red circles over here are the linear polymers. So these are the ones that were previously established. And the purple circles here are the branched polymers, which is the ones that we were making for this project. And I've simply arranged these polymers in descending order of their transfection efficiency. So polymer number 1 would just indicate that this is the best performing polymer in that class. So what we do notice is that the top performing branched polymer is able to perform better than the top performing linear polymer. But what is very important here to understand is that not all branched polymers perform better than the linear polymers. So clearly, the chemistry of the materials that you're using is important. Just the fact that you branch a polymer does not mean that you will get better transfection efficiency. We wanted to confirm that this was true for other cell lines as well. So we looked at a couple of human cancer cells. So this is a cervical cancer cell line, this is a colon cancer cell line, and we again find the same conclusion. We then looked at a pig kidney cell line, and again we see something similar. And then these are mesenchymal stem cells, and these are primary human aortic endothelial cells, which are considered somewhat difficult to transfect. And so we use them, and regardless of what cell line that we looked at, we kept getting the same results, and we found that it was the top performing branched polymer that would do better than the top performing linear polymer, but not all branched polymers would do better than the linear polymers. Now, as a side note, we were just kind of curious over here to understand whether it's the same branched polymer that does well in all cell lines, or is it that different polymers do differently in different cell lines. So over here, I'm presenting to you the same data that I had on the last slide, but in a slightly different way. So here, this is a heat map of transfection. Each row over here is a different cell line, and each column here is a different polymer. So there are about 200 columns on this heat map. The more blue the color, the greater the transfection efficiency is. So what we see on this heat map is there are certain patches on this heat map. For example, over here you see that these set of polymers actually do relatively well in all the cell lines that we were looking at. So these could be potentially used if you don't have any reservations about what cells these polymers transfect. But you see that we find patches of polymers, for example, over here, which do really very well in one cell line, but they have very little transfection efficiency in the other cell line. So one could potentially then look into whether this kind of specificity exists in vivo, and can you then use this for targeting nucleic acids to certain cell lines in vivo. One can also then sort of look at a little bit of structure activity relationship over here in terms of what branching agents are giving you good transfection. So over here, I'm showing you a relative distribution of the branching agents that went into our polymer library. So what this says is that about 40% or so of our starting library had this particular branching agent in it, the N-methyl ethylene diamine branching agent. But then if we look at just the hits, the good performing polymers in say HeLa cells, you see that nearly 60% of our hits contained this N-methyl ethylene diamine, and none of our hits contain this NNN triethylamine branching agent. So one can then, if you want to sort of move this forward and make more potent transfecting agents, perhaps focusing on these kind of branching agents could be very interesting. We see somewhat different results when we look at something like mesenchymal stem cells. So again, depending on the type of cell that you want to transfect, you might be able to do similar structure activity relationships to understand what can go into your polymer library as you move forward. So all of this work was done with plasmid DNA. We wanted to see whether something like this can be done with mRNA as well. So over here, I'm showing you some transfection data in vitro with our polymers in a colon cancer cell line, KECO2 cells. So again, we see that one of our top performing polymer over here was able to transfect about 50% of the cells in vitro at a mRNA dose of 100 nanograms. So again, this was really very promising to us. But all of this data was in vitro. Really, what happens to this in vivo? So we wanted to test whether our nanoparticles were able to transfect cells in vivo. So we first did a mouse study. So over here, we took mice and we injected them, or we treated them with our mRNA nanoparticles. The nanoparticles were either injected directly into the stomach through a laparoscopic surgery, or they were injected via the tail vein. And then we were looking at percentage transfected cells within the stomach. And perhaps unsurprisingly, we found that injection of the mRNA nanoparticles into the stomach led to higher cells transfected in the stomach as compared to when the nanoparticles were injected via the tail vein. And this goes to the access point that I was making right at the beginning of my presentation, and that obviously if you do administer the mRNA nanoparticles in the gastrointestinal tract, you will get higher transfection in the gastrointestinal tract. But perhaps what was surprising and interesting to us was also that when we did administer the mRNA nanoparticles in the stomach, we were getting some transfection in the liver as well. And this transfection was somewhat larger than what we got within the tail vein. So there was perhaps some drainage of the nanoparticles going from the stomach into the liver. And this was something relatively interesting, and it could be potentially looked at further for systemic mRNA-based therapies from an oral administration. Then we were interested in understanding what cells these nanoparticles were transfecting. So we looked at a couple of different polymers over here, and we wanted to see what cells these were generally transfecting within the stomach. And what we were seeing was that the cells transfected depended on the relative distribution of the cells within the stomach. So for example, a majority of the cells within the stomach of one kind are the parietal cells, and we did notice that our polymers were transfecting most of the parietal cells, or I should say most of the cells transfected by the polymers were parietal cells. There was a very minimal transfection within the gastrin cells and some transfection within the delta cells. But all of this was done in a way where we were actually laparoscopically injecting the nanoparticles into the stomach. And I would suspect that most of the scientists on this call are translational scientists, and you can obviously imagine that this can be difficult to do on a large scale in a clinical setting. So how do you actually then deliver the mRNA in a clinical setting on a larger scale? So in order to sort of address this question, we used a device to inject the mRNA into the wall of the stomach. Now, this device was inspired by a Weeble Wobble toy, which you might have seen children play with, or you can also say that this was inspired by tortoises that regardless of the orientation that they fall in, they always go to a certain orientation. Okay, so the idea here is that the patient actually swallows this device. It's very small, it's of the size of a blueberry or so, so this can be put into a capsule. The patient can swallow the device. Once the device reaches the stomach, it lines up in a certain orientation on the wall of the stomach, and then it injects its cargo into the wall of the stomach. So how do we actually engineer the device to do it, to do this? So this device consists of two parts. The bottom part of the device is made out of a heavy metal, and the top part of the device is made out of a lighter plastic. Now, because of the way this is engineered, it always aligns itself in a certain direction, regardless of the orientation it starts with. So this is why once the device is ingested, it will align itself on the wall of the stomach in this orientation. Now, how do we actually inject the mRNA nanoparticles into the wall of the stomach? So over here, I'm showing you a cross section of the device. So you can see that the device here contains a little microneedle. So this microneedle can be made out of mRNA nanoparticles that are lyophilized and that are put into this shape, or this microneedle can be hollow and you can fill the mRNA nanoparticles in the form of a liquid into this device. This microneedle or this I should say this needle is then connected with a spring that is maintained in its closed position. It is maintained in this closed position with the use of a sugar plug. So that sugar plug really keeps the spring closed. And on top of the device, we have little holes here. These little holes allow for the influx of water. So once this device gets into the stomach, water or gastrointestinal fluid enters into the device. The little sugar plug over here, which is keeping the spring closed, dissolves, and this allows for the actuation of the needle. The needle, as I said, can be made out of mRNA nanoparticles, or you can have something, or you can have a liquid mRNA dispersion filled into this device, and this can then be injected into the wall of the stomach. So for this experiment or for this project, we use the liquid version of the device. I should mention that. So with this idea in mind, we first wanted to make sure that this device works. So over here again, I'm showing you a picture of the device placed into someone's hand, and we first validated that this device works ex vivo. So over here, we took a stomach of a pig, and ex vivo, we actuated the device. The device in this case was filled with either insulin or it was filled with a dye to see whether we are actually able to inject the contents of the device into the wall of the stomach. So you can see that the first set of images over here are immunohistochemistry of the stomach. The top panel shows you an image at a lower magnification, and the bottom panel shows you higher magnification images. The second panel over here shows you immunohistochemistry done for insulin on the porcine stomach, and you can see that the insulin kind of is delivered across the little part of the stomach that we were looking at, indicating that there is really injection of the cargo into the wall of the stomach. And over here, you're looking at a blue dye that was injected in the stomach. And you can see over here that the injection of the blue dye happens in the submucosal region of the stomach ex vivo. So this gave us significant confidence that indeed with this device, we can actually inject things directly into the wall of the stomach. So this way you can actually protect the mRNA from the contents of the gastrointestinal tract and really directly get it in proximity with the cells that it is trying to transfect. But we wanted to see whether this is able to deliver the mRNA in vivo. We first made sure that we could actually really lyophilize our nanoparticles, get them into a very small volume so that it can be filled into the device. And then we filled these lyophilized nanoparticles, put them into our device, and we delivered it to pigs. So over here, you're looking at Western blot analysis done on the stomach of pigs that were treated with 3 devices, A, B, and C. Each of these pigs were treated with 3 devices. So each of these little points shows you a separate device that was administered to the pig. And the control animals were left untreated. So you can see here by Western blot analysis, looking for the protein that mRNA was encoding, that indeed we were able to get a protein expression in the stomach of animals in 2 of the 3 pigs that we treated. Unfortunately, we did not see any expression in the 3rd animal that we had treated with this device. This could be for reasons of us not being able to find where exactly this device deployed in a pig. As you can imagine, the pig is a pretty large animal, and looking for small places within the stomach of the animal can be relatively challenging. In addition to this Western blot analysis, we also wanted to confirm this in a different way. So over here, you're looking at immunohistochemistry analysis, looking for the expression of the protein in an animal that was left untreated or in an animal that was treated with the mRNA-based therapy. And you can see with this sort of reddish brown color that the animal that was treated with the mRNA-based therapy showed a nice expression of the protein. So again, this was another way of us confirming that indeed we were able to get mRNA delivery into the stomach of a pig with this system. We did in fact actually look for the protein expression in the liver of these animals, but unfortunately we couldn't find any. So what could be the reason for the differences that we see within the mouse studies and within the pig studies? So in a mouse, when we look for protein expression within the liver, we typically take a significant portion of the liver, mince it up, and do flow cytometric analysis on that. In contrast to that, the liver of a pig is huge. So our ability to sample the liver is actually very, very poor. So it was really very difficult for us to actually look for the places where this mRNA therapy might have gone, at least in the liver of the animal. We're looking at studies going forward where the protein that is expressed is actually expressed into systemic circulation. So then we'll be able to measure that protein a little better than what we used over here. Nevertheless, this was one of the first times people had shown that you could actually deliver mRNA into the stomach of the animal. We've started some preliminary work right now looking at what happens to the immune response when you administer these mRNA therapies in the stomach. So over here, you're looking at work done again in mice, where we were looking at antigen experienced T cells when animals were treated in the gastrointestinal tract with RNA nanoparticles, and you can see a relatively high number of these antigen experienced T cells produced with gastrointestinal delivery of mRNA. We did see some antigen-specific T cells in the spleen with this treatment, and we are seeing some antibody production as well. But nonetheless, this is early days for this field, and there's a lot more to learn as we move forward. But hopefully, just to summarize my talk, hopefully what I was conveyed to you today was that there's a significant amount of significant opportunities with oral delivery of nucleic acids, and but there are a lot of challenges associated with it. What we did for this project was we designed formulations that could allow mRNA delivery into the gastrointestinal tract, and then combine them with devices that can protect these formulations and inject these formulations in the wall of the gastrointestinal tract. And with this formulation device combination, we were able to show mRNA delivery to the wall of the gastrointestinal tract in large animals. And we're hoping to take this forward to a variety of therapeutic areas and hope to give you an update on that soon as well. But with that, I'd like to thank several of the people who contributed to this work, including our sponsors. So a lot of the work was sponsored by Novo Nordisk, and this was done when I was a postdoc, and I was supported by the Pharma Foundation. So with that, I'm happy to take any questions. Thank you very much.
Thanks, Dr. Curtin, for a very great presentation. We did receive a few questions in the panel. The first question that we have is from Rajeswari. He's asking, depending on the branching agent used, is there any or potentially any difference in the clearance rate?
Yeah, that's a great question. So a majority of the polymer still is made out of the linear polymer, which forms the backbone of this material. So we've generally seen that it's the linear polymer that dictates the degradation of these materials. We do see, however, a difference in the transfection efficiency of the material based on the branching agent that we are using.
Okay, great. The next question is from Maya. The question is, what would be the cost of such device? Is it reusable?
So that's a great question. Unfortunately, I can't tell you about the cost of the device. It's just something that's way outside my expertise is a big word, but way outside my knowledge range. So I don't know what the cost of this device would be. In terms of reusability, I don't think that a device like this would be reusable, because ultimately sort of the device will be removed through the feces of the patient. So I don't think you can reuse the device by itself.
Thanks, Dr. Curtin. The next question is from Victoria. She's wondering how much volume can be filled in the device, and what dose was used for the lipid nanoparticle delivery in vivo.
That's an excellent question. So for the experiments that I showed you, we dosed 50 micrograms of mRNA in a pig, and I showed you experiments where each pig was dosed with 3 of these pills. So the total mRNA dose that was given to the pig was 150 micrograms, but each device was able to take 50 micrograms of mRNA. The total volume that was in each device, if I remember correctly, was 80 microliters. So when we make the nanoparticles, we are unable to make them so concentrated, and that's the reason why we lyophilize these nanoparticles. And that allows us to basically modulate the volume that's going that goes into the device. So so again, each device has 50 micrograms of mRNA, and it's about 80 microliters volume. I should note that these are not lipid nanoparticles, these are polymer-based nanoparticles, and the polymer to mRNA ratio that we have used is about 100 to 1 weight by weight.
Thanks, Dr. Curtin. Yeah, I think there is another question from Wen Jen, also about the volume of the device, because I think that's one of the concerns that people have if they are actually needs high drug loading or high dose delivery. That could be a limitation of the challenge.
I think that's a very important point, and I just sort of as I said in the beginning, we work on a lot of like oral drug delivery platforms, and this is a principal challenge, a very central challenge to a lot of what we do, is that how do you put everything that you need in a system that a person can swallow. And I think in my hands at least for projects like this, lyophilization is sort of the alternative to concentrating the nanoparticles. But nevertheless, there are other alternatives too, such as diafiltration and so on.
Yeah, great. So the next question from Kanika. Since you mentioned you conducted some lyophilization study on these formulations, could you please share some insights from that research in terms of the lyophilization stability?
Yeah, that's a great question. So sorry, I rushed through this one, but I can talk about this a little more. So what we did here was we made the nanoparticles at a typical concentration that we were making it at, and then we mixed it with different volumes of sucrose solutions, and these sucrose solutions were made at different concentrations too. So here you're looking at the concentration of sucrose solution that we used, and on the sorry, I'm sorry. I think you're not seeing my screen, my pointer, but let me just bring it out here. So sorry. So here you're looking at the concentration of sucrose solutions that we used on the vertical axis, and on the horizontal axis, you're looking at the volume ratio of the sucrose solution to the nanoparticle that we used. And what we see is that really both the concentration of sucrose solution that we used, as well as the ratio of the sucrose to volume of nanoparticles, is really very important in maintaining the activity of the nanoparticles. So when we did use a high enough sucrose concentration, we were able to maintain the activity of the nanoparticles, at least in vitro. In terms of long-term stability, we actually haven't done those studies. So what happens after we lyophilize these nanoparticles and then keep them at different temperatures? We haven't done that study yet, so we don't know the answer to that question. This lyophilization study was predominantly done for volume reduction so that you can put it into the device.
Thanks, Dr. Curtin. Next question is from Yu-Hong Wu. She's wondering what might be the potential use and formulation strategy with the polymer material for oral delivery.
I guess that if I understand the question correctly, and please pardon me if I'm not if I'm misunderstanding over here. So the question pertains to, I guess, what is the use of an oral polymer-based mRNA delivery system versus something else? Do you
Yeah,
think?
I think she did not provide details, but my guess is the concern is about how acceptable that polymer material is for our delivery, since it seems to be some novel excipients that you proposed, or the synthetic polymers,
Right,
particularly for this. Yeah.
Right. No. I think that's a very important point in terms of sort of regulatory aspects. Certainly, this is a new material, so it will need to go through all the rigor of the FDA inspection before this can make it to a person. With regard to sort of acceptability, again, patient acceptability. Different people prefer different things, right? So there's certainly some people might still prefer to have an mRNA-based therapy that is either injected into them by someone else or injected into them by themselves. So this may not be for them, but there might be a certain patient population or a certain, I guess, people population who might prefer to not keep injecting themselves and take something orally. So I think with respect to acceptability, I think it's a personal choice. This is another option for administering mRNA-based therapies, I think.
Yeah, maybe a follow-up question could be, has your lab ever tried any polymers that have been used in the current drug products, such as PLGA or well-known biodegradable polymers? Do you think those are also viable options for this particular application?
Yeah, so we personally, or I should say, I personally have not used PLGA for this application. All of my PhD was done with PLGA-based polymers. They work excellently for small molecule-based therapies. I have personally not tried to use it for this application. However, I should say that there has been work done towards using PLGA just by itself for nucleic acid-based therapies from, I believe, Mark Salzman's lab in Yale. And there has been some work where people have used polyethyleneamine, which is the polymer that I showed you before, mixed in with PLGA for nucleic acid therapy. I think it's been done for siRNA-based therapies. So there is certainly a preclinical research precedence to doing this. I personally have not used it. I suspect that the device by itself can be used for other types of nanoparticles as well. It doesn't have to be just this type of nanoparticles.
Okay, thanks. I think we are right on time. We still have a few questions in the line. So for people who would like to stay, please feel free. I'll just continue the discussions from the panel. So the question from Matt is, what method did you use to prepare nanoparticles? Could you please share more details about the process itself, and also the size distribution, surface charge, loading capacity, etc. about your nanoparticle systems? I can definitely see that question from the formulator. Yeah.
Yeah, no, that's a great point, and thank you for asking this question. These are things very close to my heart, but sometimes time doesn't permit me to share all of this. But nevertheless, these polymers were prepared, or excuse me, these nanoparticles were prepared simply by complexation. So we have the polymer dissolved in. It's initially dissolved in DMSO. It gets dissolved in an acidic buffer to ionize the polymer. We complex it with the mRNA simply by mixing it with a pipette. I would think that this might get better if you have microfluidic channels or microfluidic devices to do that. In terms of the size of these nanoparticles, these range from 100 to 250 nanometers in hydrodynamic diameter as measured by dynamic light scattering. This depends on the type of polymer that we are using. It changes a lot based on the polymer that we use. Some polymers that don't work well give you really high particle sizes as well, and they generally don't work very well in vitro. Okay, so that's particle size. Surface charge, it's almost all cationic surface charge. These are unlike the ionizable lipids that give you sort of a neutral charge at neutral pH and then become cationic at an acidic pH. This is not how these things work. They're positively charged, which is probably the reason why in this data set that I showed you, these polymers essentially transfected the first cells that they saw. So again, I hope this answers the question around the formulation. I should add that this work that I presented to you today is published in a paper, and we have all of these details in there too. So please feel free to access that as well.
Thanks, Dr. Curtin. The next question from Wenjen asking, what would you think are the next steps of this device? Will frequent use of the device be an issue for the patient?
Yeah, it's a good question. So we are starting to look at applications with this system. So what I showed you today was a very, and as I said initially at the start of my talk, it's a very really a proof of concept of what we could do with this system. It's just showing that we can deliver mRNA and we do see protein production. The next steps is really applying this towards the opportunities that I mentioned to you at sort of the beginning of the presentation. Even improving these sort of the delivery efficiency of the nanoparticles by themselves is something that we are looking into. With regards to the second question about what happens when you have repeated administrations of this in a patient, I do not know the answer to that question. There are other applications of this device that are being currently studied in patients today. So hopefully with those clinical studies, a lot of information will become clear.
Okay. Thanks, Dr. Curtin. The next question is from Kanika, and he's asking, are there any other excipients used for RNA complexation?
So I think that the question is to do with the formulations that I use. So no, so we just use the cationic polymer, and then for lyophilization we use sucrose, but we didn't use any polyethylene glycol or anything in that. Yeah.
Okay. And then I guess you mentioned in your process you use the DMSO, but that's just a processing agent that will be removed later, right?
Removed, yeah.
Okay. Okay. For now, I did not see any new questions coming in the panel. Just want to do a final check. Are there any comments and questions from the audience? Okay, if not, I think we are already 7 minutes past 12. And I do not see any questions, so I think maybe we can end today's webinar. Before we close, I would like to thank Dr. Emilia Curtin again for a very great presentation. It's a very new area for us, and we're all very excited to see more new research work in this area from you and also from your colleagues. Hopefully, in the near future, we can take this technology into the clinic to the patients, and eventually maybe commercialize it to the market, so we can really benefit the patients. Okay, and I would also like to thank all the people who attend today's webinar, and for people who submit your questions. Very nice discussion. Also would like to thank the IQ committee who helped organize this webinar. And as we mentioned in the beginning, the slides as well as the video recording of today's webinar will be shared with the participants later after today's webinar. With that, thank you all. We'll close today's call. Hope everyone have a good rest of the day. Thank you.
Thank you very much.
Thank you. Bye.